Okay, so hi again. After we just finished part 5B for this session, and just before moving to the assignment video and to the exercise itself, I am adding this extra session that I called part 5C, simply because we decided to make you see a bit how these NGS files are generated practically. So there will be no exact demo for that, but we will be showing you some basic command lines that are used for classical NGS pipelines and some key steps of it, and we will be giving you also an idea about how to deal with these files easily once they are generated and given to you by your sequencing platform. And we will make you work a bit then on one specific file, which is a VCF file, using the command line interface. This will be in the video of the assignment, and it will be useful for the last part of your assignment. So, we just wanted to let you know that we just finished to see in the last video an overview of some of the most classical file format that you can use while performing an NGS experiment, right? So what we would like you to understand in this part is basically that the same basic syntax can be useful to manipulate NGS files. So I just want to make you realize that you need to be careful about that, because for NGS analysis you will be using other specific commands that do require specific installations that are obviously beyond the scope of this session, but because they require some algorithms and tools to work. But we just wanted to show you here some examples of these commands and how easy the pipelines for NGS analysis can be once you understand the basics of the command line interface. And we will be seeing some examples of these command lines to generate or retrieve data from NGS files. And we will be finally seeing how even with a basic knowledge of the Linux command interface you can still interrogate big files coming from NGS analysis platforms. So, remember that the idea of using the command line interface is just because you have too much data that is generated in a single file to be able to interrogate these files using classical tools such as Excel, for example. So, the output files are very large. These output files contain various kinds of information that you need to pass. So FASTQ will not only contain the sequence, but will contain also quality associated to each read. A SAM or BAM file will be also adding a quality to that mapping, the CIGAR string, many kind of information. And the VCF will not only show you what are the variants, but can also annotate the effect that these variants can have on the gene and on its protein. So, the command line environment is able to deal with multi-user needs, and this could be very, very useful if you have to handle, for example, multiple files at a time. So it brings a lot of flexibility to handle large files, and it allows you to easily pass the content of a normal file. So obviously, it will also allow you to pass the content of very big files that are contained in NGS. So, the basic commands you have seen are useful for NGS. Okay, we've seen that. But remember that many kinds of different files are generated throughout the NGS analysis pipeline. These files have different requirements, they have different extensions, but at this stage you should be able to know that knowing the file system's basic should allow you at least to work with these many files and classify them by creating directories and moving through file systems, because this is very important, because as I told you, you will have many files that are generated at each of the steps of the analysis. So you should be able to store them properly. And you should be able also to hand easily queries to pass these large files. So you should be able to search for a particular pattern, for example, and this is particularly the case of a grep command that should allow you to, if you remember it well, to search for this particular pattern. We will be seeing that in a moment. And you should also be able to select specific information columns, and this is specifically done with the cut command. and this should allow you, this very basic command, to interrogate at least the large amount of information in some of the output files of the NGS. So you've been seeing with Dr. Amel Rouila in the Linux session some of these commands. I'm just mentioning some of them just as a reminder, but you need to go back if you need more information to go back to that Linux session. So I wanted to mention the cat command to view the content of a file, the more, which allows you to view the content of a file step by step. Less is also useful to view it by portions. Head would allow you to view the first lines of a long file, so by default it would display the 10 first lines, but you can of course specify the number of lines you would like to display. Tail is the exact same thing, except that it shows you the last lines of this file, and by default again it will display the 10 last lines. And a very useful command to extract specific fields from a file is the cut command. So we will be seeing with the example of the VCF file that you will have different fields. Each of these fields will contain again an information of it, so you would have different what we call separators in a big file. You have separators that separate between the different fields, and you have separators that separate within each of these fields the different information that is contained. So the cut command allows you to specify the field separator that you have in your file. So here, for example, this is a space, and you can also specify the field that you would like to extract, which is in that case field number 1 and 2. So you've seen all that, you should be remembering it. grep would allow you to search for a specific occurrence. So let's say that you have a XXX pattern that you are looking for. grep would allow you to extract it easily from file 1, or you could also choose not to display this specific pattern, so everything that is not this specific pattern, using the -v option. So there are many different options for the grep. You can see the man for this command if you need so. Another very useful command is the word count that allows you, using the -L option, to print the line counts, which would be very useful. We will be seeing that in a moment. And you can obviously use the pipe character that allowed you to combine two different commands. So the output of the first command will be redirected here. So here, instead of displaying everything on your screen, this would transfer, if you'd like, the result of this command, the output of this command, as an input for this, and you would only have displayed here the word count. And you can also use this greater than character to redirect to another file. So basically the command line syntax is this one. So you have a command, you have options, and you have different arguments. And this is exactly the same thing. We will be seeing it using the different algorithms and tools with the NGS pipeline. So let's see what's happening for some of these files. We'll be only covering some of them. So you remember that we had to run a FastQC, which is a quality control on a FastQ file. Now there are many ways you can run a FastQC. Obviously you need the FastQC to be installed on your computer. We will not be covering this, but there are many plenty of resources on internet that would show you how to do that. And FastQC will generate an HTML report. The interesting thing is here that to see is that if you run the FastQC, you can do it on just one command line for different kind of files. If you have different kind of files, you have a help for this command that would allow you to see different options. I'm just showing two here, where the -O with the output and a directory that is specified will create all output files in that specified directory that needs to be created. The extract option would uncompress the zipped output file, for example. These are just examples of how to deal with the FastQC. So I just wanted to show you how easy the process is, provided that you have read the full documentation for that command and also the others. So if you remember well, the FastQC was generating the HTML file with a specific display of the reads that are of low quality. So if you want to trim this low quality basis from the the rest of your analysis, because they can cause otherwise mappable sequence not to align. There are different open source tools that can trim off this reprime basis. I'm just showing the example of one of these, which is FastX toolkit. And you have here an example of just one command line that allows you to unzip your file of interest and to pipe the result to FastX trimmer, specifying specific options, saying that I do not want, I want the last base, to be the 50th one. And this is just to say this is the specific base quality encoding that has been used, and then you can redirect everything to another file. Now, there is also other tools that are can be used for trimming adapters. This is very important because, otherwise the sequence the other sequences may not align. So you have here a way to ask for that specific pattern to be removed from this file, and the result should be output in that new file. And you have also another example showing that there are many options that could be related, and you have the explanation of it if you want to go further into the details of it. Now, a way to work with SAM and BAM files is to use SAM tools. So you have here examples of how to convert a SAM to a BAM file, which is SAM tools view, and you can sort and index these files. So one thing I wanted to show you here is that usually you use a very intuitive way to name your file. So a test.sam would be converted into a test.bam. You need to specify it in order to make sure you know what is the file you are dealing with. So you can sort, you can index these files. You can also view certain portions of these files. So here, for example, you have the chromosome 1 entirely displayed into that other specific file, or just portions of that chromosome 1. Another very useful command is the flagstat that allows you to do some metrics on your SAM and BAM. And another thing is to obtain the VCF file. At this stage, it is important to know that the VCF have to contain the information about variants. So a first step would be to call these variants. But these VCF files can also be used as input and output files for many other tools that would allow you to annotate these variants. So the way to annotate that is done by some tools, like SAM tools, for example. These tools compute statistics basically to know the different reads that share a mismatch from a reference, the sequence quality data, the expected sequencing errors, and they combine everything in order to know if you are seeing a mutation in a VCF file, if this mutation is just due to a sequencing error or to a real SNP. So a way to do that, I'm just showing an example, there are many others, is done by two steps using SAM tools and BCF tools. You have the explanation of it. The idea is just that a first step would allow you to use SAM tools, MPileup, with many different options. In order to generate first a BCF file, you have the explanations of these outputs of these options. In here, the idea is that you take a genome, with the FASTA sequence of that genome, make some different options, use some different options. Use different files you have and pipe everything to the BCF tools. You have the explanation of the different options in here. And then at the second stage, you convert the BCF into VCF. Again, this is just done using one simple command line, and you have many options you can use in here. So now a way to annotate is using JANOVAR. So you have here a way to ask for a specific refseq. This command would allow you to obtain the refseq.ser file, and then you use it in order to annotate that VCF file into that specific file. So this is very important to know, and the output would be just an effect associated to these variants. So at this stage, you should be able to know the commands, And you should be able to handle this, and this is what we will be handle all the information that is there, and you should be able now to go